Welcome to our fourth and final video for Chapter 12. We're going to look at pleiotropy, sex-linked traits, epistasis, and polygenic inheritance. Most genes have multiple phenotypic effects, and this is known as pleiotropy. So one example of this is in cystic fibrosis, where there's a mutation in the gene that encodes chloride channels in the body. So this is Cl. What kind of phenotypic effects this has on the body includes things like coughing, much more mucus in the sinuses, in the lungs. It can also result in fatigue. You're not getting as much oxygen into the blood. There could be pneumonia, shortness of breath. So multiple types of symptoms, multiple phenotypic effects due to one faulty allele or mutation in that gene. Another example is in sickle cell anemia. This is also a recessive trait, just like cystic fibrosis is. But what happens in this case is that you will have a mutation in the gene that encodes hemoglobin. So you produce abnormal hemoglobin, a protein in your blood that binds to oxygen. These cells are sometimes destroyed. They don't live as long. They're misformed and they don't bind oxygen properly. This can cause a lot of pain in the body, chest pain. It can cause problems with the spleen, even stroke, sometimes jaundice or yellowing of the skin, the eyes, the mouth. So we can see multiple phenotypic effects as a result of a single gene, a mutation in a single gene. In my previous examples, such as for the cystic fibrosis or sickle cell anemia examples, the mutant alleles were recessive. But that's not always the case. Mutant alleles can also be dominant over the other phenotypes, including the wild type phenotype. So this might occur when the mutant allele somehow interferes with a normal gene, so that even a heterozygote with a single wild type allele still expresses the mutant phenotype. In the example from our book, the mutant allele causes the expansion of how the normal gene product is distributed, and it causes the fruit fly to develop legs where usually the antennae are. In all of the previous examples, we've been looking at genes that were on the autosomal chromosomes. The autosomes, these are chromosomes 1 through 22, pairs 1 through 22 in humans. When we perform reciprocal crosses for the previous examples, it doesn't matter who the mom or the dad is in terms of which alleles they donate. You get the same ratios for the offspring. But we also have some traits that are on the X or the Y chromosome. And when that happens, this is the 23rd pair of chromosomes in humans. We call these sex-linked traits. Our Drosophila, these are fruit flies, probably the most well-studied model organism for genetics. They also have sex-linked traits, including eye color, which happens to be on the X chromosome. And our book gives us some examples of these different types of eye colors they have. Unlike the autosomal crosses, when you're crossing individuals with sex-linked traits, it matters which parent, the mom or dad, is carrying the trait. Additionally, when you look at the offspring, the ratio for the phenotypes will usually differ when you look at males versus females. So let's look at this one more closely. We have a dad, and I know that because of the symbol, but also because of the Y chromosome. And he has a big W on the X chromosome, which is the wild type phenotype, the red eye. The mom is recessive for both of the alleles, which is the white eye phenotype. It looks like they're putting mom's gametes here. Gametes are haploid, so that's why you only see one allele above each box. And dad's gametes are over there. Dad's can either give an X chromosome or a Y chromosome. And the trait over here for eye colors on the X chromosome. If you complete the Punnett square and look at their offspring, it looks like all 100% of females will have red eyes. Why is this the case? So to be female, you inherit an X chromosome from dad. So both of these X chromosomes are from dad, the one with the big W. So they have to be red-eyed because they get the red eye allele from dad. The second allele they get from mom, and mom has two of the same alleles, so that's a small w. These are all heterozygotes. If I look at the males, males have to get a Y chromosome from dad to be male, and they have to get an X chromosome from mom, and that happens to be a small w. So 100% of the males have the white eye allele. So this was the previous problem, but our book then gives us another problem that we can try. What ratio of offspring would result from a cross between a white-eyed male? So let's say this is the male and it's white-eyed. Males are XY, small w. Female is this symbol. Sorry, ignore that. And females I know is XX. She's heterozygous for red, so that would be big W, small w. And I want to look at the offspring, so I can make a Punnett square. I'll put mom's gametes on the left. That's X big W, X small w. Dad's on the top. And if I put them together, I have a big W, small w, small w, small w, and then big W, Y, small w, Y. These are the males on the right, and I see half the males will have, let me change colors, half the males will have red, and half the males will have white. And these are eyes. So a 1-to-1 phenotypic ratio and genotypic ratio for males. For females over here, the same is true. They're going to have a 1-to-1 ratio of red, this one's red, to white eyes for females as well. And I wanted to introduce you to a new term. We've seen homozygous and heterozygous, but what about males? Since males only have one copy of the X chromosome, and let's say they have an allele here, that's not homozygous or heterozygous. We call this hemizygous. Hemizygous, because they only have one X chromosome. Hemizygous. Let's switch over to humans from fruit flies, and we are going to see similar effects. So let's use the letter A for the trait where big A is normal. Let's say dad is normal, he's unaffected, so let's say he's big AY. And mom is unaffected as well, but she's a carrier. So that means she has one abnormal allele and one of the disease allele, and they are on the X chromosome. So the son of a woman will have 50% chance of being affected. Why is that the case? I know because sons will for sure get a Y from dad, and they have a half a chance of getting either X chromosome from mom. That's where the 50% comes from. What about daughters? So daughters, remember the dad determines the sex of the child. So daughter, meaning that she got the X chromosome from dad. Now the one that she gets from mom is also a 50% chance of getting this one or this one. And notice how for carriers, they half shade it. They shade it halfway. Sometimes you'll see that on pedigrees as well. Besides alleles that cause disease, we also have lethal alleles, alleles that are deadly. And these can be dominant or recessive, but luckily the dominant lethal alleles are very rare. One example from our book are Manx cats. Manx cats are heterozygous and they have a shortened tail. The lethal allele is dominant in case this is dominant. Homozygous cats that are recessive for the allele, homozygous recessive for the allele, these are normal cats. If you have two copies of the dominant lethal allele, the cat will not survive. So if you have two Manx cats, you're going to produce a ratio of 2 Manx and 1 normal. So let me do that. We have our Punnett square. So I get a normal cat, a heterozygous, another heterozygous down here, and then a homozygous lethal cat. 2 Manx to 1 normal cat. That's the offspring ratio because that is lethal. That cat will not survive. You're going to see a 2-to-1 ratio of offspring. So technically an allele only has to shorten the normal lifespan to be considered lethal, and this is usually because it won't be passed on to the next generation. The most common lethal allele example for humans is Huntington's disease. Huntington's. This is a dominant lethal allele, and what happens in Huntington's is the nervous system starts wasting away. If someone is heterozygous for this disease, this lethal allele really, they will eventually develop the disease. But Huntington's has a late onset, so you might not start seeing symptoms until you're in your 40s, and that means that the allele may have already been passed on. So your children have a half a chance of getting the lethal allele. Earlier in Chapter 12, I gave you an example of organisms where we're examining 3 characteristics and calculating the probabilities of their offspring. In our book, they mention that when you're looking at trihybrids, let me simplify by just using the letters A's, B's, and C's. This is a trihybrid organism crossed with another trihybrid. They mention that you can use the fork line method, as shown here, to calculate the probabilities of the offspring. But you can also use the multiplication and addition rule, the rules that we mentioned earlier, multiplication and addition rules. In fact, this really comes into play or comes handy when you have more alleles than shown here. So maybe you have another letter, so you're looking at 4 or 5 or more characteristics. Where do trihybrid organisms come from? Because usually when we start with a parental generation, we're looking at true breeding organisms that have, for example, all dominant alleles or all recessive alleles. One way is through crossing them, or we would get a trihybrid organism. But another way is through crossing over. That happens during prophase 1 of meiosis. We saw this before in the previous chapter. So this is another way to get different types of alleles from true breeding parents. Earlier we talked about pleiotropy, with one gene having multiple phenotypic effects. We can also have genes that affect the phenotype of other genes because of how they interact. And in one case, epistasis, that's exactly what's happening. In polygenic inheritance, in contrast, we're having multiple genes that independently affect a single trait. And I'll go over that example afterwards. Let's start with epistasis. Epistasis means one gene stands over another gene. So the expression of a gene at one location on the chromosome will change the phenotypic expression of a second gene at a different location on the chromosome. So I like to give an example that is not really true. It doesn't happen, but it's easier to understand. So let's say A tells you if you have hair, big A. If you have small a, you have no hair. And the allele B, if you have big B, you have brown hair. If you have small b, you have blonde hair. So I know anyone with at least one big A is going to have hair, and then the second set of alleles will tell me what color. So this one will have brown hair, and the second one will have blonde hair. But if I look at 3 and 4, they're not going to have hair. So it doesn't matter if it was going to be brown or blonde because they both have no hair. So in this example, the gene or the allele A is epistatic or stands over the allele B. Our book also provides an example of epistasis using coat color in mice. And in this case, the C allele is going to be the epistatic gene. So C will be dominant or epistatic and stand over A. So it looks like A is a goody coat color. So A is a goody. And small a is black, and that's recessive. That's a recessive coat color. And C tells you if you have pigment. Big C has pigment, one of these pigments. Small c, there's no pigment, and that would be albino, so no coat color. So usually what we see for a dihybrid cross is that 9-3-3-1 ratio, right? 9 of them are going to show both dominant phenotypes. 3 will be one dominant, the other recessive. 3 is recessive dominant, 1 recessive recessive. Oops, sorry, AC. That would be the phenotype. But whenever you have epistasis, instead you're going to see a 9-4-3 ratio or 9-3-4, depending on how you arrange it. You won't see the 1. They'll be combined. Let's see why is that. So whenever they have big C, let me switch colors. I know the coat color will appear. So this is agouti on the left, the 9. This is going to be the black coat color. So 9 agouti, 3 black. But these two have both small c's. That means there's no coat color. No coat color, so it doesn't matter what the coat color would have been. These are both going to be albino. So 3 plus 1, that's where you get that 4. 4 are albino. What about polygenic inheritance? Earlier, I mentioned polygenic inheritance is when multiple genes independently affect a single trait. So usually you see this when you see characteristics that vary in a population along a continuum. What does that mean? Something like height, right? We have all different types of heights. Some of us are 5 feet 2, 5 feet 2.5, or someone is 6 feet 1 inch tall, or 5'6.2. So there's a continuum, and that usually indicates some kind of polygenic inheritance. At least 2 or more genes are involved in generating that phenotype. And one example is what I mentioned, the height. There are over 180 genes in humans that affect the height of a human. And another good example is skin color. This is controlled by many different types of genes as well. And this can be further complicated by the fact that some traits are not only genetic, but also depend on the environment. And when this is the case, we call these multifactorial outcomes. So this is true when we have traits that depend on both multiple genes, but there are also environmental influences on the phenotype that you see. And one of the most common examples of multifactorial effects are hydrangeas. So their flower color depends not only on their genes, but also on their environment. When they're grown in basic soil, they turn pink. But if you grow them in acidic soil, they appear this bluish, sometimes violet color. That takes us to the end of chapter 12. In chapter 13, we're going to look at modern understandings of inheritance, including linkage, linked genes, and additional disorders that are due to an abnormal number of chromosomes.